flattering introduction, so I do appreciate that. Can I just start by thanking all the local organisers, in particular Nadima and Mahmoud, for inviting me here, not just to see your university, but to get my first taste of this beautiful country. We're loving it here. So I thought I'd start off by showing you our far from beautiful city of Leicester. And here it is. It's a very typical Midlands town right in the middle of England. The only good thing I can tell you about are two things. First of all, Leicester City Football Club is now in the Premier League by a miracle. And secondly, it is, I think, one of the most successful multicultural cities anywhere in the world. Very shortly, we will have a majority Asian population. And if you travel around the streets of Leicester, you'll see not only churches and town halls and so on, but you'll see a wonderful display of Hindu temples and mosques. Okay, so it's a very interesting city, and it really does work. However, I'm not here appointed by the Leicester Tourist Board to tell you about the joys of Leicester, but instead to celebrate with you 30 years of forensic DNA. It was 30 years ago that we first, completely by accident, got involved in the concept of using DNA for identification purposes. So what I'd like to do today is to really tell you the story of forensic DNA, how we got into it by accident, where it is now, and where it might go in the future. Now, the younger people here will probably think, well, the role of genetics in forensics started with DNA, and that is absolutely not true. We can trace the history of forensic genetics back 112 years to this guy, Carl Landsteiner, the discoverer of the ABO blood group system, the first simple Mendelian character to be discovered in humans. And in a landmark paper in 1902, he speculated that maybe you could use ABO typing on blood taken from a scene of crime to try and help identify or exclude the criminal perpetrator. And that was a brilliant insight and led directly to a huge field of science, which is now completely extinct because of DNA, the field of forensic serology. So if I was giving a forensic talk 30 years ago, I wouldn't be using the term DNA at all. It was not in the game. Instead, I'd be using this bizarre language of different blood groups, the ABO blood group, rhesus, MN, secretor, and so on. And these are the tools that geneticists used back in those days to try and identify crime scene samples, to try and establish relationships, if they existed, between different people. Now, if you go back in time and talk to those people, say in the mid-1980s, about forensic serology and these types of genetic markers, they would have told you they were fantastic. They were the best in the world. They could really crack cases. And that simply wasn't true. These marker systems had serious limitations. Lack of discrimination, very restricted range of different human samples that could be typed using this sort of approach. And all of these limitations, with the wisdom of hindsight, you can get over that by looking not at variation in the products of genes, which is what you're doing here, but by instead looking directly at variation in the genetic material itself, in DNA. And we had to wait 75 years before that became possible. So, I'm going the wrong way here. There we go. So if you go back, say, 30-odd years, what could you discern about the human genome, the totality of human DNA? Not much. It's stored away in 23 pairs of chromosomes in each cell, and those are the chromosomes, as seen down a light microscope. If you take those chromosomes and look at them under an electron microscope, I hope what you can see is what looks like a huge, impossibly gigantic pile of spaghetti. You see the tiny strands here, this enormous complexity of the human genome. And it was only in the mid to late 1970s that the technology started to appear which enabled scientists to break into that tangle, pluck out single pieces of human DNA and study them in detail. And at that point, we started getting our first insights into two questions. First, how is information stored in human DNA in terms of storing information in the sequence of bases along the double helix? But secondly, and for me equally interesting, was how does that DNA sequence vary from one human to another? Unless you've got an identical twin, you are a genetically unique individual. So how is that uniqueness expressed in terms of variation in DNA sequences? And in 1978, both my own group and a group in California showed, using very primitive techniques of the time, that you could directly study variation in DNA at that most fundamental level of all, variation in the DNA sequence. Okay? So, and the sort of variants that we were finding were variants like this, where as you go along the DNA sequence code, most of the letters or bases, the 4 different letters that make up our genetic code, don't vary from person to person, but occasionally you'll find a position here which might be an A in some people, a C in other people, or you might find some people that have inherited A from one parent and a C from the other. And that was really exciting. We could show that there were millions of these different positions of variation scattered around our genome. But the tools at the time for accessing this variation was very limited, and equally bad, there was poor discrimination. So, for example, a single site like this can be used to divide the whole of humanity into just 3 groups. Those that have got A from both mum and dad, those that have got C from mum and dad, and those that have got an A from one parent and C from the other. So lack of discrimination. So we then started thinking, around about 1980, that surely in that immensity of the human genome, there must be bits of DNA more variable than these boring little single letter variations, what we now call single nucleotide polymorphisms, or SNPs. And initially, based purely on hypothesis, we conjectured that maybe as you go down the DNA double helix of humans, you might find the occasional region where a short sequence of DNA would be repeated over and over again in tandem. So it's like stuttered DNA. And if such regions exist, then they'd have the possibility of varying, not in the sequence, but in the number of stutters at a given position. So you can imagine a chromosome carrying maybe 5 stutters, or a different version with 8, 10, 15, 23, 42, 122, 793, endless possible variations. In other words, highly variable DNA. And why we were interested in getting at such highly variable segments of DNA? Simple answer, basic tool for human and medical genetics. Mapping the human genome, mapping genes involved in inherited disease, and as an aid for diagnostics. No forensics, nothing at all. Okay, we call these regions mini-satellites for reasons that I don't need to go into. So the question is, how do you identify, if they exist, these regions of DNA? So let me just remind you, short sequence, maybe about 30 bases long, repeated tens or hundreds of times. And initially we had no idea. And then 1 or 2 examples were discovered by accident, by some good colleagues of mine, so we knew they existed, but we still had no general method for getting at them. And then we got lucky. So the story, in fact, of DNA fingerprinting and forensic DNA starts with me, a seal, and the British Antarctic Survey. This is the start of forensic DNA. Why the British Antarctic Survey? Because we wanted some seal meat to study some very obscure aspects of gene evolution. We're particularly interested in a gene called the myoglobin gene that is expressed at very high levels in the muscle of diving mammals, like whales and seals. So if you're going to look at that gene, you need to get hold of muscle from one of these creatures. And locked away in the British Antarctic Survey headquarters was a freezer full of lumps of whale and seal meat. So I went there, they gave me a lump of seal meat, It looked about the size of a house brick. They also gave me a recipe for cooking it. So if there's anything left over after the experiment, you could turn it into a curry. And quite honestly, looking at that seal meat, I thought, no, that is an experiment too far. I'm not going to try cooking it. So anyway, it worked well. We got the seal myoglobin gene. Then we got the human myoglobin gene for comparison. And inside that gene was one of these stuttered bits of DNA, mini satellites. But curiously, we found out fairly quickly that that mini satellite could cross-hybridize too. It could detect very weakly other mini satellites in human DNA by virtue of making weakly matched Watson-Crick double helix pairings between this mini satellite, used as a probe, if you like, and target mini satellites in the human genome. In other words, it looked like different mini satellites. Even though they had different repeat sequences, there was some sequence shared in common. And when we defined what was going on, it was a very simple story. The different mini satellites tucked away inside the repeat unit was this little patch of red DNA, about 15 bases long, that was shared almost identically between different mini satellites. So different mini satellites at different positions in the human genome can share this sequence similarity. What is this sequence doing? Even today, we don't know for sure. We've got some ideas. But it told us practically how to get at mini satellites. You simply make yourself a hybridization probe, a detector if you like, which just consists of this little red sequence repeated over and over again. That should be really good at sticking to lots of different mini satellites. So at that point, we thought, great, we can now isolate different mini satellites from human DNA and then use these in medical genetics and so on. But we thought, before we do the isolation, we'd better check that that idea is right. If we take this DNA and react it against human DNA, we should see lots of variable DNA fragments. So let's do the experiment. And what did we get? Well, it's a good job we did the experiment, because what came out of that was what proved to be the world's very first DNA fingerprint. So what do we have on here? We had my technician, her mother, and her father. You can tell all three individuals apart in what looks like a bit like a barcode that you see on supermarket goods. So we immediately spotted, purely by accident, spotted the potential for DNA-based identification. Also, we could see inheritance here. So, for example, this character in the child has come from dad, this has come from mum, and so on. That immediately suggested family relationships tested by DNA. And across here, we have a series of non-human species. Unfortunately, I sold the notes and this autoradiograph in charity in auction. So I can't remember what sample's what. I forgot to keep a copy, but I think that's a cow, I think that's some sort of monkey, and I know for sure that is tobacco DNA there. So it looked like these highly variable patterns, these DNA fingerprints, you could get them not just from humans, but from a very wide range of different higher organisms. So that was a very exciting moment. Within a few minutes, my entire life had changed. When did we get that first DNA fingerprint? I can tell you to the minute. 9:30 on the morning of Monday, the 10th of September, 1984. At 9:10, we were thinking forensic thoughts big time. But an important point here is that 5 minutes earlier, at 9 o'clock, I didn't have a single forensic thought in my head concerning the possible use of DNA in catching criminals and so on, and nor did anybody else in the world. So you get your technology first, then you start thinking of applications. So it was a very exciting 10 minutes for me, I can tell you. So we were then faced with two huge challenges. First, those patterns are horrible, agreed? They're all fuzzy and blurry and so on. I mean, there's no way you'd take that to a court of law. So we had to improve the technology, and that worked very easily. Within a couple of months, we were getting these very rich and informative patterns. These patterns are genuine DNA fingerprints, single test, a unique identifier with the sole exception of identical twins. So you can see just how variable these patterns are, each of these characters being a discrete mini-satellite in the human genome. Now, the variability becomes even more impressive if I tell you that these are not unrelated people. This is a single family with dad present twice and his 11 sons and daughters. And you can see a very simple pattern of inheritance here. So each character in dad is transmitted at random to half of his children. Here's a different one to half of the children, okay? The other half don't get them. Now note that we don't have mother. We don't need mum. Throw her away, she's irrelevant. We can reconstruct her. So for example, we have a strong band here in 3 of the children, absent from dad. Where's that come from? Mum. Yeah, this is not rocket science, this is very simple. So by a very simple subtraction process, you can build up essentially the entire DNA fingerprint of a person that you can't even get at. No problem. Now, with patterns like that, we knew we had something of real practical utility. Big question now, would anybody ever take any notice of this strange new technology? And if I've done one smart thing in my life, it was calling this DNA fingerprints. If I'd given this a more suitable name, like idiosyncratic southern blot mini-satellite hybridization profiles, it would have killed it dead. But calling it DNA fingerprint got the message out. This is DNA and identification. So we published our first paper on this in March 1985, expecting there to be months, years, possibly decades before implementation. Wrong. Next month, we're taking on the first case. And what was the first case? Was it a murder case or something like that? No. It was an immigration dispute with serious doubts about family relationship. It involved a young boy who was believed not to be a true member of a family living in London and was facing deportation. And we were asked by the family lawyer to come to his aid. They'd been through blood group testing. It said the boy was related in some way, but the British authorities thought that he might be a nephew to the family, not a son in the family. So here's the very first case. This is the very first DNA case ever to be tackled. So let's see what we got. And this was just a month after the first publication. Here's a mum, here's a boy about to be deported, 3 undisputed children, and the interpretation is extremely simple. Look at this boy. That band comes from mum, present in one of the other children. That band from mum. We've got bands down here from mum. We have a band here, absent in mum, present in 2 undisputed children. Where's that from? Yeah, well done. It's from the missing father. We don't have the father here. It was simply not available. In fact, the mother thought there were 2 possible fathers to make things even worse. Okay? So coming down here, every single genetic character in that boy can be ascribed either to the mother or to the missing father. Conclusion, he is a full member of that family. Chance that he is a nephew to the family is vanishingly low. We're talking about certainty levels here, round about 99.999999% certain that he is a son, not a nephew. Case closed. So we then submitted that evidence to the British authorities, who then immediately dropped the case against that boy. So that was the first case ever tackled, and what a fantastic conclusion to the case. Young lad reunited with his family, and all the authorities and government and so on thoroughly embarrassed by this new technology, DNA. So that was a great start. It was shortly after that, in the summer of 1985, we took on the first paternity case, which then unleashed another wave of interest in DNA typing. And then we had 2 years of insanity where my lab was the only lab that could do this. So it was a huge relief in 1987 when this went commercial and we opened Cellmark Diagnostics, still going strong today, who provide a commercial outlet to DNA fingerprinting. And it's interesting to look at the profile of their casework over the first 10 years after their opening. And the initial bulge was in immigration disputes. So starting in 1987, year on year, quick climb in the number of immigration cases being taken on, and then they die away. Why do the numbers die away? Because there are only so many families trapped in disputes where the question was about family relationships. And we simply cleared those families through the system. That's why the numbers died away. Then with a lag, then the big explosion was in paternity testing. And what I would like you to look at are the numbers up the side here. These are thousands of cases per year. So here was a technology that within just a year or two of that academic laboratory invention was reaching out and touching the lives of thousands upon thousands of people. And in a very profound way, finding out the paternity of your child, bring immigrant families back together again with DNA. Those are life-changing information that you're giving to families. So this is quite humbling as well as exceedingly exciting. So those are some of the applications on the human side, but there are non-human applications as well. So conservation biology. Let me just give you one example from the world of conservation biology where DNA fingerprinting came to the rescue. Wall-drap ibis, highly endangered. I think there are only about 20 breeding pairs in the wild left in North Africa. Most of these birds are kept in captive colonies, the main one being in Zurich in Switzerland. But in a captive colony, you run the risk of inbreeding, of close relatives breeding. So how do you know whether these birds are breeding with close relatives or not? Well, you could sit there and watch them 24 hours a day, 7 days a week. And even that, they're quite sneaky. They'll probably do things without you knowing. Or you can get blood samples from the entire colony, do the DNA fingerprinting, which is exactly what Esther and Signa in my lab did, and those are the DNA, beautiful DNA fingerprints of every single bird in Zurich Zoo. And you can then, by looking at those, you can build the pedigree structure of that colony. So the colony was founded by 6 birds brought in from the wild, and we can now answer with this rather complicated looking pedigree whether anything dangerous is happening. And the answer is, it most certainly is. So we have mother bird and father bird here. These were founders. They got together and had 4 offspring. This brother and this sister then took a fancy to each other, and they have 4 of their own offspring. Full brother-sister mating, massive inbreeding, genetically extremely dangerous. Much more of that colony could have gone to extinction. So with that information, you simply tell the zookeeper, they get all these bad birds well separated from each other. You break the colony up into smaller colonies, and you keep genetic diversity going. So there's a real practical application. Another application is in the area of wildlife forensics. And again, this is one example from Esther Signa from my laboratory. This magnificent bird is a golden eagle. It's a CITES-listed species, pretty endangered. A man was found with an adult male, adult female, and 3 young birds. And he said he bred them himself from the 2 adults. And anybody who knows anything about ornithology will tell you that that is impossible. You cannot breed these birds in captivity. Therefore, they were stolen. So the DNA fingerprinting was done, and here are the patterns, some really nice DNA fingerprints. And conclusion, that man was telling the truth. The adult male and female were the genuine biological parents of those 3 fledglings. End result, they prevented a criminal prosecution. This guy probably would have gone to jail, and instead I hope he got a medal from the Royal Society of Birds for achieving the impossible and keeping this species going. Now you might think wildlife forensics, that's very specialized. Well, think again. And I've been to so many forensic meetings where this wildlife forensics gets ignored completely. So how big is wildlife crime? Believe it or not, it ranks 3rd in the world, right up with human trafficking in terms of total monetary value. It's beaten by only 2 other criminal areas. That's drug trafficking and the counterfeiting of goods. Wildlife and environmental crime is right up there, as I said before, with human trafficking. and way ahead of small arms trade, the illegal small arms trade. So make no mistake, wildlife crime is huge, and DNA can and will play a very big role in this. For example, just one concrete example, they are now databasing the DNA of every single rhinoceros, both living and as museum specimens, in an attempt to put some sort of halt on this insane trade of rhino horn, particularly into China, which is threatening to take rhino species to extinction. So you can now track by DNA a sample right the way from the crime scene source in Africa all the way through to a dealer wherever in Eastern Asia. So this is big and this is important. Now, what I haven't said is anything about human criminal activity. And the reason for that is that patterns like this really are too complicated to take to a court of law. The lawyers will run rings around you. You've got to simplify it. And we knew that right from the outset. So if you're going to get into human forensics, you've got to make the pattern simpler. And we were doing that anyway, because our original goal was to take human mini-satellites and isolate different ones from the human genome, which is what we did, and we started doing that about the time the first DNA fingerprints were coming off. And what that enables you to do is to, instead of looking at many different mini-satellites at the same time, to produce these very rich but complicated DNA fingerprints, you can look at just one mini-satellite at a time to look at variation in just one section of DNA. And if you do that, you get what we call DNA profiles, much simpler patterns on X-ray film, where each individual here is represented by two different bands. And you can see how variable these patterns are from one person to another. And you might say, why two bands? Because you have two different versions of that mini-satellite, one inherited from your mum and one from dad. So again, you can use these not just for identification purposes, but also looking at family relationships. Again, we felt that it would take many years before that sort of evidence would be ever accepted in police investigations and in criminal or crime scene analyses. Again, I keep getting it wrong, completely wrong. The first clone mini-satellites were coming off the production line, I guess mid-1985. By the end of 1985, early 1986, we'd taken on what proved to be the world's first criminal DNA investigation. And it was a terrible case involving two young schoolgirls in the county of Leicestershire, where I live, both of whom had been raped and murdered. And following the second murder, a young man was arrested and was charged with and confessed to that second murder. The police said that the patterns of the crimes are so similar that they're sure that whoever carried out the second murder carried out the first. So we were given the appropriate forensic samples. I expected to get nothing out of this. Nobody had ever tried this before, but it worked. So these are the results, some of the results we go back to the police. So if we look at the first victim, so this is her DNA profile, and this is a key forensic sample consisting of semen and vaginal fluid recovered from the pubic hair of that victim. And in there you can see her DNA profile, plus another two bands up here. That is the two-band DNA profile of the assailant. And only two bands, meaning that semen was from one man. If it had been from two men, there would have been four bands, and so on. So we then look at the second victim. These are trace amounts of semen, so the DNA profile is faint. I hope you can see two-band profiles here from the semen. Look very similar to the two-band profile over here from the first victim. So the semen derived from both those victims was from the same man, almost certain. So the police are right. The same man had indeed raped, presumably murdered, both of those girls. Was it the person in custody who confessed to the second murder? No. This is his DNA profile. Complete mismatch. So what was going on here? Well, after a lot of agonising, we decided, yeah, the science is okay. That was a false confession. So for the first time ever that DNA was used in anger in a criminal investigation was not to prove guilt, but to prove innocence. And without that DNA evidence,
I have no doubt that that young man would have been convicted and would have gone to jail for the rest of his life, and the true murderer would have gone free and free to commit assaults again. So what the police then did was to believe completely in DNA and to try and get blood samples from every single male of the right age group within the local community. A DNA-based manhunt, which they did, and that eventually, and I don't have time to go into the whole story, it unmasks this person, Colin Pitchfork, as a true perpetrator. So he was identified, the police went to his door, arrested him on the spot, he immediately confessed, and he's now serving 2 life sentences for those murders with a minimum tariff of 28 years. That was the birth of forensic investigations using DNA. Within a year, DNA profiling has swept right around the world, everywhere from Canada, the United States, all the way to Australia and New Zealand, we're doing DNA profiling. And it remained the technology of choice right into the 1990s. But there were limitations. It's slow. It wasn't very sensitive. It took a week or 2 to get a result. It chewed up lots of DNA. There had to be a better way forward. And the solution to that big problem came from what by any measure has to be one of the strangest characters in the story of molecular genetics. And it's this guy, Carey Mullis, who had one great idea in his life, allegedly on an acid trip while driving with his girlfriend in California. And he's published actually very, very little, but I think the message, actually this is a very mixed and dangerous message. I'm not suggesting young people here should go and get stoned out of their heads and have a brilliant idea. But what it does say is that if you can come up with one great idea in your life, you will get a Nobel Prize, even if you do very little subsequently. And that's what happened to Carey Mullis. So his idea was very simple. DNA replicates. One of its jobs is to carry information from a mother cell to the 2 daughter cells. It's got to do that by copying the DNA. And of course it does that by separating the two strands of the double helix, and then you copy, so you go from a molecule of DNA here to 2 molecules. And what Carey showed was first that you can do that in the test tube, and secondly, if you go from 1 molecule and copy it to 2 molecules, there's nothing to stop you from separating the 2 molecules into the 4 strands, copying again to make 4 molecules, and again to make 8, 16, and so on. And of course, what he invented was DNA amplification using technical term calls as the polymerase chain reaction. As soon as the user-friendly version of that appeared in 1988, we knew that was going to be the way forward. It offered speed, it offered sensitivity, it offered the potential for automation. And it also offered the potential to get away from these great big mini satellites. These are bits of DNA, thousands of bases long, that all too often are broken up in an old forensic sample, and instead we could move to much shorter microsatellites and simple tandem repeat regions, which are again repeated. So this one just reads GATA or GATA, repeated over and over. So that sequence is just GATA, 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 GATA. The number of GATAs will vary from one person to another, and the number of GATAs can be assayed simply by amplifying across this region and measuring the length of the amplified bit of DNA. The more GATAs there are, the longer it is. Simple. And these are tiny, so these are perhaps 100th the size of a mini satellite, and they will survive in an old forensic sample. So we saw that pretty well straight away, and then attempted to deploy this in what proved engaged in one of the first investigations ever to use PCR or microsatellite typing. And that was a really important case, a case that would have been absolutely impossible using DNA fingerprinting technology. And that was the identification of the remains of this person who's arguably the most notorious individual war criminal ever to have existed on this planet. It's Dr. Josef Mengele, the angel of death, the Auschwitz concentration camp doctor, that, amongst many other perversions, carried out experiments on live victims, particularly twins, in, for heaven's sake, the name of genetics. This is a terrible person who evaded the Allies at the end of the Second World War, escaped to South America, and then someone who might have been him drowned in a swimming accident in 1979, exhumed in 1985, and the really important question for many governments and institutions around the world was, were these remains those of Mengele or someone else? So we were faced with the tricky task of taking remains that had been dug out of a grave in Brazil in very, very poor condition, trying to extract quantities of DNA from there that proved to be down in the 1,000th of a part of a millionth of a part of a gram of human DNA. That's how much you could get out. And then getting a microsatellite profile and comparing that with living relatives. Unfortunately, we had access to Mengele's wife and son, who were both living in Germany. So what we had here was a bizarre paternity case. A very, very macabre paternity case. And these are the results that we got back. It proved to be pretty straightforward. So, rather strange looking DNA profile. These are microsatellite DNA profiles. Each band has some shadows underneath, which I'm sure you can ignore. So, if we look at the wife of Mengele, she has 2 bands. This band has been transmitted to her son. This band is therefore from the father, and these bone remnants have got that genetic marker in them. So that result is consistent with the remains being those of Joseph Mengele. The paternity test works. But by itself, that's not enough. In fact, we had to use 10 different microsatellites and show that every one gave the same answer before we could be statistically sure, at the 99.9% level, these remains were those of Mengele. And that did work, and that allowed probably the biggest war criminal investigation ever mounted against an individual that had been dragging on for 40 years to be brought to an end. But all the authorities, along with a lot of other evidence, accepted that these were indeed the remains of Mengele. So that was a very high-profile case. Let me just give you a few other of my favourite high-profile cases that we did not do over the subsequent years. A brilliant case was that, anybody recognise these good people? Yes, Tsar Nicholas Tsarina, murdered by the Bolsheviks in 1980. Remains of them and their three of their children exhumed in 1985 in Western Siberia. But the big question was, is that really the Tsar Tsarina and their children, or some other Russian noble family? So in a brilliant analysis, the Home Office Forensic Science Service looked at the skeletons in the grave, went through microsatellite typing and said, yes, there's a genuine family group here, mum, dad, 3 children. And then used a very special DNA called mitochondrial DNA that's only inherited down the female line to tie those remains in to living maternal descendants of the Tsar and Tsarina. So, for example, the Tsarina's sister's daughter's son is our Prince Philip. Just remember, all the royal families of Europe are all tied together. So, if the Tsarina's sister's daughter's son was Prince Philip and the remains were those of the Tsarina, they should share the same mitochondrial DNA type. And they did. And likewise, on the Tsar's side, the Tsar's mother's daughter's son or something, some guy living in Germany, he was typed, and again there was a good match. So, conclusion, those really were the remains of Tsar Tsarina and their children, and the end result was a state funeral in St. Petersburg for those remains, 80 years after the Bolshevik murder. Wonderful case. In terms of high publicity, the top one had to be the case of O.J. Simpson, American sports superstar, charged with the murder of his ex-wife and his ex-wife's boyfriend. That went to a 9-month trial where in the States there was, I'm not exaggerating, there was an O.J. Simpson TV channel that broadcast this stuff 24 hours a day, 7 days a week. This got DNA into the living room of every single home in the United States. That got DNA out to the public. And of course he got away with it. He was acquitted in that trial. I wonder why. Well, I'll let you speculate about that. What about the world's naughtiest case? Easy, very, very easy. That has got to be the case of President Bill Clinton, Monica Lewinsky, and the interesting semen stain on the dress acquired during presidential activities in the Oval Office. And again, it was a raft of DNA technologies, including our DNA probes, that we used to tie that sample in with Clinton. And other slightly more recent cases of huge importance, of course, the identification of Saddam Hussein and Osama bin Laden. When both were caught either dead or alive, key question, is this really them or some lookalike, a bodyguard? Big question, certainly in the case of Saddam Hussein. So in both cases, DNA typing results comparing with living relatives came in exceedingly rapidly. In both cases, in 24 hours of their capture, there's verification of identity. So, of course, the technology has evolved over the years. So we go back to the Mengele case. These are pretty complicated, stuttery, Stone Age patterns, I like to call them. And what's happened over the subsequent years is that technology has been simplified. So the stutters have been gotten rid of. You don't look at one microsatellite at a time, but 10. And you color code the amplified bits of DNA by putting very pretty fluorescent colors into the amplified DNA. So a modern looking DNA fingerprint would look like this. So here we have 10 different microsatellites. So here's a green one up here, and a yellow one there, and a blue one, and another green one, and so on. So 10 different microsatellites, and a sex test here, which will tell you whether the sample is from a male or a female. Very powerful technology, very robust. I mean, first implemented in the mid-1990s, and has really not changed a great deal since then. So this is a so-called SGM Plus system, which is the basis of all typing within the United Kingdom. Now, if you take a pattern like that, so each person has their own distinct pattern, that's pretty complicated, but of course you can scan that and convert it into a much simpler form. So for a given individual, for each microsatellite or STR, all you want to know is how big are these bands, how many stutters are in them. So your first STR, you might have one with 10 repeats from one of your parents, and one with 12 repeats from the other parent. And the next one might have a 9-14, and the next both are 8s, and so on and so forth. Now, it doesn't require a genius to spot that a table like that is a very simple digital code for an individual, and what you do with it, you stick it in a computer, and you make yourself a nice computer database. And the first country to deploy that concept was the United Kingdom in 1995, where the then Home Secretary launched our National Criminal Intelligence DNA database, using what are 6 at the time, but now 10 microsatellite markers, that give you tremendous discrimination. If you want to take 2 of you at random, the chance that you would match is about 1 part in 10 trillion. But for close relatives, it's much more likely. And in fact, this is not enough markers to be really safe. You need to increase that. Now, the sex test is in there, and originally the database was conceived for a databasing of all people convicted of a criminal offence, so that if they re-offended, they'd be quickly identified by DNA and brought to justice. How big is that database? Well, the latest figures from 2012 is almost 6 million people are on that database. That is 10% of the entire UK population now resides on that database. Impact on criminal investigations, enormous. So DNA is now the technology of first resort. You go to the crime scene, you look for the DNA evidence. If you find the DNA evidence, you check that profile against the database to see if you already have got the perpetrator databased away. And that, in a huge number of cases, has enabled suspects, its crime scene samples, to be matched to a suspect on the database, and equally importantly, finding matches between different crime scene samples. For example, a serial rapist leaving the same semen but on different victims at different geographical locations. So there's no question, this is a very important police tool. Does it have its downsides at all? Well, there are some problems. I'll just give you one, which has now been addressed, I'm pleased to say, and that is in Britain, in about 10 years ago, the law was changed which enabled the police to take a DNA swab from anybody they arrested, irrespective of whether that person would subsequently be charged with a criminal offence or even prosecuted. Just arrest was enough to get your DNA on the database for the rest of your life, without appeal. And the end result of that was 1.5 million entirely innocent people on the database, and with a huge ethnic bias. So about 80% of African juveniles, or people of African descent, juvenile males, were on that database. So the government was basically branding an entire sector of society as future criminals. Massive discrimination. That was declared illegal both by the European Court of Human Rights and subsequently by the UK Supreme Court. And it was only last year that those samples started coming off the database. And they're not all off yet, by any means. So the battle's not quite over. What about other countries with databases? Well, this is just a quick snapshot of the whole world. Dark red means they've got a national DNA database. Pink means they're going to go for it. So they're in the process of doing it. So basically, it's virtually the whole of Europe, North America, Australia, China, and so on. A very strong uptake in North Africa and the Middle East. And sadly, there's a grey patch right slap bang in the middle there. That's Turkey. All right? I know you're doing DNA forensics. I know you're sticking your DNA on a computer. For heaven's sake, get a national DNA database together. To not do so is criminally irresponsible. You will fail to capture criminals by not doing that. Okay, so that's one message. Okay, what about the sizes of these databases? Well, there's no way you can read this, but this is just Central and Western Europe. A database size vary enormously. So major countries, Italy hasn't got a database either. So don't feel too bad, Turkey. You're not the only ones not to get there yet. And then most others have got, in terms of convicted people, in the tens of thousands, maybe hundreds of thousands, until you get to Britain with nearly 6 million. In fact, if you add up all the people that have been databased, 55% of them across Europe are British. Now, Britons are not responsible for 55% of European crime. No matter what you think about us, we're not that bad, honest. Okay? No, it's different countries have very different criteria for taking and retaining DNA. And the British government has been very, very aggressive in getting as many people into that database as possible. So that's where we are at the moment. You might ask, how many people worldwide, how many criminals worldwide are now on these databases? And the answer, we don't know for sure, but there's an awesome, at least 50 million people worldwide. That's approaching 1% of the entire world population is now sitting on a database. And if that's not a sobering thought for technology that didn't exist 30 years ago, I don't know what is. So just a few other, I've got a few more minutes? Yeah. So just a few other little aspects of forensic DNA. So database is brilliant for catching criminals, but remember our first murder case where we showed innocence? That's an equally important aspect of DNA testing. And that's best exemplified by the Innocence Project from the United States, where they systematically champion the post-conviction testing of DNA evidence to see whether the conviction was secure or not. And to date, we've got over 300 long-term prisoners in the United States released, entirely innocent. And I'll just show one of them. This is someone I've actually met. This is the first person ever to be exonerated by DNA. So that's him and that's me. This is an award ceremony in London. Never mind what it was. This is Kurt Bloodsworth. He had been convicted of the rape and murder of a 9-year-old girl in the very year that we came up with our first DNA fingerprint. He was sentenced to death by gas chamber in the United States. It went into one of the worst penitentiaries in the United States. And as part of this punishment, each month he had to go into the gas chamber and clean it. Civilized country? I'll leave you to work that out. Anyway, he saw the potential way forward as DNA. That was his hope. And the DNA testing was done, and it was shown that he was entirely innocent of that crime. So that man was literally saved from death by DNA, quite literally. And that was a very emotional meeting, I can tell you. And if you want to know more about that story, it's all been written up, not in a boring scientific paper, but in this book, Bloodsworth. The true story of the first death row inmate exonerated by DNA. And it's got a twist at the end that you really couldn't make up. And I'm not going to tell you what it is. You've got to go away and read it. Suffice to say, it would make a brilliant movie. Shawshank Redemption with DNA is probably the best way of describing it. So that's, I mean, real human impact. Real human impact. So, other aspects of DNA. Mass disaster analysis. Twin Tower attack in Manhattan in 2011. 25,000 body parts recovered, over 1,000 successfully identified and returned to their families by DNA testing. The mass war graves in Bosnia. The International Commission for Missing Persons is now systematically getting skeletal remains out of these graves, DNA tested, and cross-checked with the entire local population to make sure that, again, the remains of loved ones are returned to families to help bring closure. And of course, that approach is highly relevant to Cyprus as well. There are exactly the same issues here. Perhaps not on the same scale as Bosnia, but equally painful to the local populations. Also, mass disasters. The Asian tsunami, 300,000 people died. That saw the mobilization of virtually every forensic DNA laboratory in the world, faced with that gigantic task of identifying individuals, many of whom are eventually washed up to decompose for conventional identification purposes. And what about the future of forensic DNA? Well, one possibility. Let's suppose you go to a crime scene and you get yourself some DNA. You've got yourself a DNA profile, and you look on the database. There's no match. So you've got someone who hasn't got a record. Okay, so who is it? Well, wouldn't it be nice to look in the DNA and get their name out? Now, you're all thinking, Jefferson's gone completely insane. You cannot get your name out of DNA. Well, stop and think carefully. How are surnames inherited in most countries? I think it's true in Turkey. Inherited down the male line, is that correct for Turkey? You inherit from your father. Am I right? Hello? Yeah, same in Britain, okay. What else is inherited strictly down the male line? The Y chromosome, the chromosome that carries the male determining gene. So you've got surnames in the Y chromosome faithfully co-transmitting generation to generation. What that means is that in theory there should be correlations between the surname you carry and the Y chromosome that you have. Right. Has anybody heard of David Attenborough? Yeah, the wildlife guy on television, right. People have tested this. For example, if you get as many Attenboroughs as possible, it turns out that most of them share exactly the same Y chromosome. So that tells you something very interesting. First, that the Attenborough name had a single evolutionary origin, probably about 600 years ago. And it also tells you something very interesting about Mrs. Attenborough's, that they've been very faithful to their husbands. Because if there hadn't have been, you wouldn't get that link between the surname and the Y chromosome. You get wrong Y chromosomes coming. Agree? So you can actually measure the rate of historical non-paternity using this approach, which is what I said before. So that's a wonderful approach, and of course the flip side is if you find that Y chromosome type at the scene of crime, you may not know who it is, but could be an Attenborough. And that is an incredible lead for the police. Unfortunately, for most surnames, this doesn't work. Now, as far as I know, this has never been tested in Turkey, and who knows, you might find absolute gold dust here, or maybe even bigger flop than the UK, but it's not the general rule. So what about other possibilities? What about taking a DNA sample and trying to reconstruct the physical appearance of an individual from variation in that DNA? The concept of a DNA identikit. Well, there's some stuff we can do. As I've already shown you, we can do sex of an individual. It's actually not a very good test. It's a test that scores 5% of Sri Lankan men as women. That's not very good, is it? That's a standard test. Hair colour, red hair is a really good test. For other shades of hair, it's getting there, but it's pretty iffy. Eye colour's good, 95% accurate prediction of the shade of blue or brown eye or green that you have. Skin colour is getting there. Stature, how tall you are, very, very difficult. Over 500 different genes control your height. Variation in those genes. So even if you can crack that, it's going to be such a complicated mess that making an accurate prediction of the height of an individual, I think, is going to be probably impossible. Possibly theoretically impossible. Facial appearance, even less progress there, but there is a little bit of a start there. Look, also face is what we all associate with inheriting. You know, you've got your mother's eyes and your father's ears, we all know that. Yeah, how does that work at the DNA level? No idea. Absolutely no idea at all. Earwax consistency, that's a good one. Very simple test. You've either got sloppy or stiff earwax. How you use that forensically, I have no idea. Ethno-geographic origin is a really big one. That's a whole lecture in its own right. But suffice to say, there are batteries of markers that can identify the ethnic origin of an individual and about the continental level, but it's getting more and more fine-grained now. So within Britain, there are some very sophisticated tests that can even distinguish at the county level. You're talking about distance of a few tens of kilometres. You can pin someone into a region. But again, that will not be of general applicability. You can even figure out someone's age, roughly, from a blood sample. And with some development of tests, I think it might even be possible to say, go to the crime scene, here's a blood sample, we don't know who it's from, but I can tell you that whoever left that blood sample left it at about 3 o'clock in the morning, or they left it around about 2 o'clock in the afternoon. You could even predict the time of day at which that blood sample was deposited. There's a photo. Okay. So why do the police want to do this? Well, because they haven't got the person on the database, they're trying to get some other link. Is there another approach? There's a very obvious one that will work, and that is put everybody on the database. Have a universal database, everybody included per country. Then you will always get it. You will always find what's bad. Always, and women commit crime as well occasionally. So, is that being done in the UK? No way. Civil liberties issues are gigantic. Is it being done anywhere? Yes. Right now in the United Arab Emirates. They have legislation enacted that it will make it mandatory for every citizen and every guest worker to be databased in that country. However, the progress is very slow. The focus at the moment is on criminals and on the military. But they have the potential to put the entire database on the population. Why do they want to do that? Well, they say, well, it's to help in crime. Well, most of the crime in Dubai is in fact fraud, financial crime. Okay, where DNA comes in there, I have absolutely no idea. Also mass disasters. Well, when was the mass disaster in Dubai? Or UAE? I can't remember what. Could it be state surveillance? No. Didn't hear that. Okay. So I think they've embarked on a social experiment with potentially very dangerous outcomes. Let's see where it goes. And I'd just like to finish off with a challenge for the future, the new people, the next generation of molecular geneticists. The best you can do in terms of getting a readout is about 20 minutes from getting your crime scene sample to getting a DNA profile. If you can get that, that's a long time. If you can get that down to a few seconds, you're then into the world of real-time security applications. So, for example, as you enter a country, use DNA as the ultimate biometric. So, quick saliva sample, very rapid test, you are with what your passport supermarket. So instead of putting your credit card and then your PIN in, put your credit card in, and then you spit on something. They said, yeah, that's fine. You take it. Can it be done now? No. Is it theoretically impossible to develop such fast systems? No, it will happen. The challenge is for someone to figure out how to do it. And I just want to finish with one last slide, which is one application I haven't covered yet. This is an application relating to people who in Britain rent out, or came from people renting out holiday cottages. So this is a tourism problem. And I hope this isn't a tourist problem that also affects Northern Cyprus. Maybe it does, you can tell me. But this is not only an example of a very powerful potential application of DNA, but a very powerful argument for the universal database. And with that, I will finish the talk. Thank you. We're interested in, well, actually, dear Sarah Mann, I'm not sure whether I'm male or female. We're interested in genetic fingerprinting devised by Leicester University as owners of self-catering accommodation, as holiday cottages. We wonder whether this method could be used to verify the persons responsible for leaving urinated beds. We will close the stamped address envelope for your. That's sweet. And of course, DNA most certainly could be used to find these vile perpetrators. To my knowledge, thank you very much indeed.
Are there any questions to Professor Alec Jeffries?
I can indeed, yes, thank you.
I understand that you invent the key.
We can't hear. We can't hear.
I can repeat, don't worry. Yeah.
I understand that you invent a key to open the tennis part, the goddess of the justice. And you said this was accident. I wonder if Newton's apple falls down just in front of Newton, accidentally.
Right. So this is a very flashing question. I think I'm being possibly likened to Isaac Newton, which is great. Actually, Isaac Newton was a really horrible man. Did you know that? Nobody liked him. He was a great guy, a great scientist. Human being. He was absolutely dreadful. No, the question was that we had developed a key which opened the door on a technology that would impact on justice, and that was this really an accident or not? And I think, well, the answer is absolutely yes, it was an accident. And if you look at the, a lot of the big events in scientific history, and whether these are planned or not, in many cases they weren't. So Isaac Newton, I suspect he was certainly mulling over the issue of attraction between objects. And I think the apple, it gave the right sort of trigger in his mind. In the same way that Einstein was happily travelling around on trains and realised, well, actually, I may be moving fast, but I feel as if I'm not moving at all, and it looks like the world outside is moving fast. And it was that social context and the technology context that started getting me thinking about the whole concept of relativity. And then if you look at some of the inventions, I mean, did Alexander Fleming deliberately leave that Petri dish open by his window to allow a penicillin spore to land on his plate and start secreting this thing which he then called an antibiotic? No. The thing that all these people, I think, share is accident followed by just keeping your eyes open and seeing what's happening. That's the secret. So, for example, the microwave oven is another good example. the microwaves were first developed as a communication device, and then someone had a microwave in the States, had a microwave transmitter on one side of the room and a receiver on the other side of the room, but they left a chocolate bar, a Hershey bar in the middle. Now you know what happens with a Hershey bar in an oven, right? So they switched on the microwave transmitter, there's an almighty explosion and a vaporized Hershey bar. And they thought, oh yeah, we just invented the microwave oven. Same with the laser. That the graduate student that developed the first laser was told by a supervisor to stop it. There's no practical consequence to a laser. And of course every home, every car, everything you can think of has got a laser in it. So I think the are two messages here, or several messages. First, accidents do occur in science, and the important thing is have your eyes open to spot those implications. Secondly, that you cannot plan for a lot of these advances, and that is an argument for making sure you maintain a very vigorous fundamental science base driven by curiosity, not by the desire to solve a problem. I mean, you know as well as I do that if there's going to be a revolution, for example, in the treatment of cancer, the initial key to that, it's not going to come from an oncologist. No way. It will probably come from a botanist or something like that, who will come up with a concept which you never even thought of, which can then be translated into practical research. So you've got to keep the fundamental science going, and that is a really hard sell at the moment, because all governments are strapped for cash. They want to be assured that the pounds and the dollars and the Turkish lira and so on they're putting into research, they're going to get back threefold over in terms of the economy. And you can't guarantee that with basic science. It doesn't work like that. All you do is to scatter as many seeds as possible, securing the knowledge that a few are going to germinate and produce something fantastic, and the rest are going to die. So be it. Yes. Sorry, that was proselytizing. Yes.
Thank you very much for your talk. I think to summarize what you just said, your pastor once said, a lack favors prepared mind. So you're prepared to do discoveries.
So Louis Pasteur quotes about luck favouring the prepared mind. I think that is right. So if you get a wacky looking result, look at it.
Yeah. I work for a missing persons from Cyprus, and thanks to your contributions as well, we have made 500 identifications so far, Greek secrets and Turkish secrets, using DNA profiling.
Yes, indeed. Yeah, it has worked. I think what we haven't seen yet, correct me if I'm wrong, is a sort of integrated national effort. It's a little bit piecemeal at the moment. I think that the problem with doing identification from grave sites is having that reference population. So basically, you need a properly scaled organisation to do that. It's not something you do in a piecemeal way. So it is working, but I suspect it could work better. And of course, for every family involved, they want to know. So these are, I've sometimes said, it's tried, but there have been, heaven knows how many millions of cases worldwide that have been tackled with DNA. Let me say, every single case is a drama for the person involved. So this is not something trivial happening. For that individual, it's a drama. And some of the most traumatic ones are the loss of loved ones, whether through a natural disaster or a man-made disaster. So remains identification enormously important. And actually quite a strong argument for you getting your DNA profile onto some sort of secure database as a future insurance against disaster of whatever sort for identification. Yes.
Just want to ask a quick question about mutations. What mutations actually theoretically somehow lead to false positive identification in some cases, or actually would that be not the case?
Right. So the question is is mutation a problem in, for example, creating false positive identifications? Well, the answer is no. If you have, even if mutation were a major problem, mutation will make identical patterns different, not different patterns identical. That's extremely unlikely. So you can worry about mutation problem at that level of discrimination below the level of 1 in 10 trillion discrimination. But the other issue is that when you take a DNA sample, say, from the scene of crime, you're doing consensus testing. You'll be looking at maybe 100 cells worth of DNA. So within those 100 cells, if there's a mutation in there, maybe one cell's got it. So what you see is the 99%, not the 1%. So where it does become an issue is when you are doing extremely sensitive targeting down to the level of a single cell. Then that can become an issue, and much more important in paternity testing. So it's certainly the case, even going back to DNA fingerprinting, that mutation from the outset was an issue. But it's an issue that can be readily addressed. Very occasionally, I have a paternity case where you get into a grey zone, that, yeah, there's a couple of mutations here, we're not sure if it's paternity with mutation or non-paternity, but the true father is a close relative of the alleged father. So he's not the dad, it was his cousin, brother, or whatever, it was the father. So yes, I mean, with any technology, you'll always come up with the occasional grey area case, and you report it as such. But these are rare, so not a problem.